Good evening, good afternoon, wherever you are in the world. We welcome you to our ISTA-ISSS series of webinars, the last before Christmas. And I've got two speakers here today on the topic seed vigor, mechanisms and applications. It's Alison Powell and Gerhard Leutner. Before we start, maybe the information that this webinar will be recorded and will be made available on the ISTA YouTube channel in a few days. Sometimes it's even there in the evening, depending how quick it can be put together. We will have only one question and answer session, but you are allowed to put your questions into the question and answer box. And we only use this box to answer the questions in the end. So let us start with our first speaker. I first welcome Gerhard Leutner. He is Chair of Plant Biochemistry at Royal Holloway University of London, RHUL. Has more than 30 years research experience in seed germination and seedling establishment of crop, wheat, and wild species. Maybe we have to say that there's a wild species working group or committee now in ISTA as well, who just recently launched a new website, and you can find that via the ISTA website. He obtained his PhD at Ruhr-Universität Bochum in Germany with a doctoral work at the Ruhr-Universität and Swiss Federal Institute of Technology in Zurich, Switzerland. Gerhard leads RHUL's Seed Biology and Technology Group, a team conducting world-class impactful research using multidisciplinary seed-focused approaches into food supply chain security and sustainability in collaboration with their international partner network in academia and industry, including Syngenta, KWS Seeds SE, TOSA Seeds, Royal Botanic Gardens Kew, and the European Space Agency. I will have a question a little later, Gerhard. What are you doing in the space there with the seed? And he is corresponding author of the highly cited Tensley Review on Seed Dormancy and Germination, New Phytologists 2006, and curator of the Seed Biology Place website, which is www.seedbiology.eu. And in 2021 was awarded the prestigious Alfred Meyer Plenary Lecture by the International Society of Seed Science. So we have a very important man here, and I hand over to you, Gerhard.
Thank you very much, Andreas. I share my presentation and hope that everybody can hear me. So the first presentation is about more the fundamental aspects of seed vigor and seedling performance. And what you see here on the slide is Royal Holloway University of London's founders building. It was in the Victorian era one of the first women's colleges, which is quite a remarkable history, which I want to mention. Today, we have a seed biology and technology team. This is led by Tina Steinbrecher and by myself. You may have met most of my colleagues in the conference in Australia. Who was not there was Anna, our newest member. She joined just a few weeks ago. And so this is the team which does the work here. And we do this, as Andreas already mentioned, in collaboration with industry and academic partners, and by funding, especially from the UK research councils, but also from the European Space Agency. So seed vigor is intrinsically connected with climate change, abiotic stresses. And without going into details, this slide shows the component heat and drought extreme impacts. So it's not only a little bit of warming, which is a problem for many crops, but an advantage for some, but it is the extreme weather which is the most difficult to deal with. And for many crops, an example here is sugar beet, the predictions are that yields are affected by these changes. This extreme weather is flooding and waterlogging, drought, heat, but also frost damage due to early drilling, and technology, breeding and priming technology are technologies which are applied to improve this and reduce the stress. So at the bottom, I usually have some literature. As you get the slides later, you may pick some of them for reading if you like. What I enjoyed reading before the seminar is part of Friedrich Nobbe's book 1876 in Samenkunde. And this book describes the Keimung, the germination process. And I have copied this part here and will read it shortly. Keimung ist diejenige Periode der Entfaltung des pflanzlichen Embryos, in welcher die formbildenden Aktionen von den im gereiften Samen aufgehäuften Stoffen der vorausgegangenen vegetativen Arbeit der Mutterpflanze entstammt. Eine Metamorphose ohne Stoffbilde. Essentially what this tells, and this is also interesting in this book, is that germination was seen at this time as the entire process from the imbibition to the emergence of the seedling. And this is also evident from what is called in this book, for example, hypogeal germination, which you see here for pea. At the same time, Julius Sachs was working on the effects of temperature on germination. So this is probably the origin of the works into how plants deal with abiotic stresses. Nobbe coined this term seed or seedling vigor, which is Triebkraft. It can be translated into shoot strength, driving force, early seedling vigor, and emergence tests like those by Hildner were the initial tests which were used for testing it. So if you look at pea and a pea seed, it has huge storage cotyledons and an embryo axis. And if you treat it with ethylene, what you see here on the right is that the hypocotyl and the root are shorter in the ethylene-treated pea seedling, pre-emergent seedling. And this is a reaction to growing through the soil, so for the pre-emergent growth, where ethylene plays a role, and ethylene is also considered as a vigour marker. Interestingly, ethylene is produced by the pea seed just by this embryonic axis. So the huge cotyledons do not contribute at all to the production of ethylene, and the production of ethylene is associated with the late phase of germination, where it is a transition to the early pre-emergence growth. So ISTA defines vigor today as the sum of those properties that determine the activity and performance of seedless lots of acceptable germination in a wide range of environments. And consequently, ethylene would be one of the possible markers in vigor. So seed vigor is therefore a concept, a potential performance of viable seed in agricultural practice. It's not a single measurable seed property, which makes it more difficult. And this figure here from one of my favorite reviews by Finch, Savage, and Bussell shows you what it is about. So this is different field situations. And what you always see in gray is the laboratory tested percentage of germination, which is more or less close to 100% for all these situations. But on the other hand, seedlings may get lost in the post-germination phase, and in green you see the huge variability in seedling emergence. And this is what vigour is addressing, this difference between laboratory seed germination and per se, and the in-field conditions performance in terms of seedling emergence. So vigour is on the left side during seed development, progressively acquired during seed development, and increases until the physiological maturity. And then it is later during seed storage decreasing, but it decreases first. So if you store a seed, what decreases first is the vigour, and only later the viability will decrease. So therefore, if you do a germination test, only looking at the first part of vigour, the curve, if the vigour is lost, is shifted to the right here, and later then, when viability gets lost, the seed lot also has a reduced maximum germination. So vigour, therefore, has these components: the rate or speed and uniformity of seed germination upon abiotic stress especially, so unpleasant temperatures, not optimal moisture content, and then seedling emergence under unfavorable conditions, so abiotic stresses, the soil seed bag, and vigour is therefore a trait which spans these different processes. This is an example for small vegetable seeds, also taken from this review here from Finch, Savage, and Bussell. And what it shows is that an aspect of vigour is first the rate of germination, and then a second aspect is the rate of initial downwards growth of the hypocotyl and the root, and then third, an upward growth in strong soil so until the shoot emerges, and this is in interaction with the seed bed, for which the deterioration causes that it becomes stronger in the upper parts. So we know something about this, and this has to do with in brassica, some work in brassica on seed vigour, quite some publications. And I apologize already that I cannot mention everybody's publications here. There are so many, and many really good work can't be presented here. So abscisic acid plays a role in these processes, especially in germination speed, ABA sensitivity, and ABA catabolism. But also work from the Penfield group shows that maturation temperature during development defines ABA sensitivity and DOC1, so residual dormancy, which may be expressed at these stages. So the work of Bill's group in 2010, 2016, especially using this concept of these three parts of the trait, identified in QTL screening several gene candidates, which are listed here. One is called the speed of germination, the other reduced ABA acid QTL, and two of them are also involved either in ABA sensitivity or in alternative splicing. And then there is a homologue of CYP707A, so the ABA hydroxylase, which degrades ABA. And so in this model here, which is from this publication by Morris. There is a concept which describes vigor during development and imbibition and the expression of these genes. So follow-up work from here is of course needed to further verify this. Another recent review by Reed, Bradford, and Kanday in 2022 has this image here, which connects vigor as a trait to climate change and highlights something which is very important, namely that, like seed dormancy, also vigor represents aspects of a physiological continuum that begins on the mother plant during seed maturation, reaches a peak vigor after the loss of primary dormancy, and ends eventually with the loss of seed viability. So vigor can go up to a point at physiological maturity where it's maximum, and then later, in a way, it can only go down. You can prime seed and increase the vigor, but it will decrease later. This review also has supplementary list with references and list of vigor candidate genes. So it's, if you want to know something about more specific genes, that's a source for looking at it. I want to mention two more gene candidate sets. One comes from rice, vigor in rice, but this publication here. So I found quite some interesting vigor candidate genes, expansins involved in cell wall, layer proteins in storage, heat shock, also in stress responses, and also gibberellin, response modulator protein, and an auxin gene, taking together hormones, ABA, GA, ethylene, auxin especially, are important for vigor, but also reactive oxygen scavenging, cell wall loosening, when there is seed deterioration, the repair of it, the amount of stored reserves, and any other aspects of stress resilience. I want to add a few more examples for deterioration and repair. This is selected examples. DNA damage, for example, and its repair. Protein damage and its repair. And also membrane lipid damage and its repair. And here are some example publications for which I do not have time to get into. So there are more of these studies, and again, my apology for not being able to include these into the presentation. I want to go back to this concept of the three traits, and the first seed vigor trait would be the rate or speed of germination, and for this, in this example here, it must be this stage, and this is affected by soil temperature and moisture. In this work, we compared two different sugar beet accessions, seed lots, and one of them had issues at 5 degrees. If it was not primed, if it was primed, there were no issues. And in this case, when you analyze these phases separately, so you germinate fruits at a warmer temperature and then analyze the pre-emergent seedling growth, there was no difference. So in this case, the example is that the difference in secondary dormancy induction and in seed vigor at this unpleasant temperatures, cold temperatures, is due to the germination phase and not to pre-emergent seedling growth. I will not discuss further any germination and dormancy aspects of seed vigor, because this has been thoroughly done. So I think our research in this area of germination is well advanced, but we know much less about these other phases here, the pre-emergent seedling growth. So the second vigor trait is this rate of downwards, initial downwards growth. And you may remember this image I showed with the ethylene-treated pea seedling, which has a root able to be strong and grow through soil. And in these experiments here with tomato, for example, if you inhibit ethylene signaling, then the root penetration is reduced. And there also the same effect can be obtained, not obtained by inhibiting auxin transport, but again, if you inhibit ethylene sensitivity, it is reduced. So root penetration is an issue, and this review here depicts that there's a strict interaction between ethylene signaling, auxin and pins, as well as biosynthesis of auxin and ethylene signaling for root and apicotal penetration of the soil. Other work here in rice is penetration of soil by the initial roots of compacted soil. So at the bottom you have non-compacted soil, which is easy for the wild type, also for this ABA deficient mutant and also for the auxin deficient mutant. But in the case of the compacted soil, ABA deficient and auxin deficient mutants are better and stronger in doing this. Here in this graphs is the root penetration ability, and as you can see, ABA deficient mutants are better, auxin deficient mutants. So again, here a connection to ethylene, which controls auxin biosynthesis, for example. So root soil penetration, the second vigor trait, is not only important for crop species, there is also natural variation here in the root penetration index if people analyze different Arabidopsis accessions. And going further to wild plants, these here are plants growing in the Mongolian steppe system, in a natural ecosystem. They have different root penetration abilities. So seeding adaptation to physical barriers by enhanced root penetration is a very important aspect of seed vigor. So coming back to the original novel and ISTA definition with seed vigor as a potential performance of a viable seed in an agricultural practice, I show another figure similar to the earlier one. In this case, it's about sugar beet. Sugar beets are fruits, and what you see here on the right is the laboratory germination and the field emergence compared of different seed lots. As you can see, laboratory germination is 95–100% in all the cases, but field emergence differs considerably. And this is exactly what the vigor trait tries to include, this difference between simple laboratory germination and the real field germination. Now, talking about sugar beet, you can ask the question whether all plants behave as I presented before. Are they all, do they have all the same mechanism for seed vigor? If you look at QTL candidates, which you get in the literature. There are similarities, but there are also differences. Sugar beet itself in its genome is very different to many other plants. So for example, in the number of transcription factors, it has far less transcription factors than most plants, about half of the numbers. It has about half of the ABA signaling factors compared to Arabidopsis, tomato, or rice. Also for auxin, about half, ethylene, also half. So, and it can cope with stresses. So it must do this a bit different or a bit easier. I don't know, it's not sorted, but it's an interesting aspect of stress resilience. The second about sugar beet and many other vegetable crops is that seed technology is used, namely polishing, priming, coating, pelleting, and all this technology is affecting vigor in the same way as breeding can affect vigor. The processes and also the growth of the mother plant can affect the vigor trait. So here an example for sugar beet. Sugar beet also has these three phases, but first of all here on the top I want to emphasize it's actually a fruit, a dry fruit, and the true seed and the pericarp can be separated. And if the sugar beet fruit has germinated, the root tip penetrates the endosperm here. There's an aspect for the seed technologies, which is biomechanics, the embryo growth potential, and the restraint of the pericarp and also the endosperm here is an issue and has been looked at, for example, in this review by Tina Steinbacher and work with sugar beet by my group. We looked at sugar beet germination by comparing seeds and fruits. And if you look at the seed stage, what we covered in a publication in 2007 for hormones is the first phase here, germination and the rate of germination, and also the initial part of the downward growth, the second phase. If you analyze what is happening to abscisic acid, a hormone which inhibits seed germination, it decreases rapidly during the germination process. And this is also associated with the increase in CYP707A, the ABA degrading enzyme, compared to dry seed and the imbibed seed here at T1%, it's much higher. And if you prime sugar beet seeds, then the transcripts of this enzyme are much higher already in the dry state, and consequently also the decrease in ABA is earlier and faster. What about ethylene? ACC increases in the late phase of germination and early phase of seedling growth, so this is number 2. And again, there's an increase of the ACC oxidase, which converts ACC into ethylene from the dry to the imbibed state. And if you prime seeds, it's already higher in the dry compared to the prime states. So very similar to what I showed you before. And I want to mention the work emphasized by ISTA-ISSS that the ethylene evolution of sugar beet is just starts just at the point of the completion of germination during the phase 2 here of seedling growth. But then there's also auxin, and IAA increases here. And if you treat sugar beet seedlings with 10 micromolar ACC, assume this is acting via ethylene, what you can see is that this is not, as in the case of the pea seed, inhibiting hypocotyl and root growth, but promoting it. The same is with IAA, it also promotes it. So sugar beet differs from these other species in that the 10 micromolar concentration has a different effect. It promotes instead of inhibiting it. So it seems to differ in ethylene sensitivity. And this is one of the issues with ethylene. It can have this Janus effect, that at low concentrations it may trigger, and at higher concentration it may inhibit. And the sensitivity for this may be different in different seeds. So I also want to use sugar beet for presenting research in this third phase. So we have the first phase of a trait of seed vigor, the rate of germination, especially upon abiotic stress. A lot is known about this phase on the molecular level. Far less is known on the rate of initial downward growth, the second seed vigor trait. And also less is known about the third phase here, namely the potential for upwards growth in strong soil of sugar beet and other species. Here's some work on this. This is work by Rinaldi et al. And on modeling sugar beet growth either in loamy soil or in clay soil. So the seed germination looks a bit more similar than the seedling emergence here. And I have modeled this data in that I calculated the germination rate here in black and the seedling emergence rate here in red. And what you can see, seed germination, GR20 is reaching, is higher, much higher than the seedling emergence rate at 20%, and the same is for 50 and 80%. This is at different water potentials, and if you compare this to clay soil, then you get a very different picture, and in this case both germination rate and seedling emergence rate are affected. So this is an example for different soils, growth through different soils, and this is also something which was at the original Nobbe-Hildner definition of vigor, and the early vigor tests, which were Triebkraft Keim tests with Ziegelgruß, brick grit. So they used soil and had a crust of brick grit here of different strength, and using this they found a different strength for this phase 3. So this phase 3 is, in my opinion, also very important for vigor, and I know that many companies use early immersion tests at cold temperature and so on. So all those goes into this direction, but the really mechanical part here is of interest too. This recent work here, I found this from 22. They looked at soybean seedling emergence, and they developed an assay with a defined crust-like gypsum layer to investigate the seedling emergence rate. And then they tested different soybean accessions, 82 accessions, and there were quite some differences in this emergence rate. So in summary, phase 1 here, rate of germination, loads of work there done, and we have advanced in this part. These two phases here are, in my opinion, more neglected in research, and they may be used as vigour tests, I know, but the background, the mechanisms here are less known. For the future, in fundamental research into mechanisms of seed vigour, I think it's important to tackle climate change. Aspects of the research can therefore be simply improving seeds by breeding and priming for improved crop seedlings emergence upon stress, addressing different environments, including even space flights or gardening on Mars. And then, of course, most important, unlocking untapped biodiversity in stress-resilient trait genes, including from wild species and land races. So one of my main messages is also that seed vigour is not only something for crop species, but it has also a lot to do with wild species and the competition which plays in natural environments for seed germination and seedling establishment. So that's the end of my presentation. Thank you for your attention, and later questions can be answered, which is organized by Andreas.
Yeah, thank you very much, Gerd, for your nice presentation. And there's a lot of applause coming up, which you cannot hear. You can hear me, right? Yes. There's a lot of applause coming up as well. And I now welcome Alison for the second presentation.
Yeah,
you can hear me, right?
Doesn't like it.
Can you hear me?
I can, yes.
Okay,
yeah.
perfect. Yeah, good. Then we continue with Alison. And as mentioned before, we have the question and answer session after Alison's presentation. And Alison is going a little bit, I think, more into the application of this test, the vigour testing in the seed testing area. And I would like to introduce Alison. Even a lot of people may know her. Her research is over 25 years at the University of Aberdeen, including the physiological basis of seed quality and its use in seed quality evaluation and also seed invigoration. She has had many roles within ISTA, very long list, a member of the executive committee, seed symposium convener, deputy editor of Seed Science and Technology. She's still doing that, chair of the Seed Science Advisory Group and member of the Rules Committee. And Alison was, of course, chair of the vigour committee since 2001, when the first vigour chapter was introduced into the ISTA rules, including the conductivity and accelerated aging tests. Since she has overseen the validation and introduction of 3 further vigour tests to the ISTA rules and the addition of a number of species to the 5 validated tests. She stepped down as the chair of the committee this year, but she remains as an active member and handing over the responsibility of a chair to younger colleagues. Alison was awarded a Doctor of Science degree DSc in 2004 in recognition of her research contributions to the advancement of knowledge in seed science and technology on the basis of an extensive publication record. Alison's contribution to the International Seed Testing Association was recognized in 2013 when she became honorary life member of the association. Alison, I hand over to you, and we are very excited about your presentation.
Thank you, Andreas. And hello, everybody. It's nice to be with you today. I'll get rid of my face and increase the size of the PowerPoint for you, and then we'll be able to get going. go. Okay, we're ready. Well, it's good to be here with you today to talk about the seed aging repair hypothesis as a basis for image analysis in vigour testing. And what I'm going to do today is talk about the problem of seed vigour, then how aging can be considered a cause of vigour differences, and how changes during aging can be used in seed vigour tests. I'll then focus on the radical emergence vigour test and the role of metabolic repair before we come to the aging repair hypothesis. Then we'll move on to the germination progress curve to the actual RE test. We'll then look more to the future, look at the potential for image analysis and molecular methods. So we'll start off with seed vigour. What is the problem? Well, if we think about what the physiological aspects of seed quality are, the most important one is germination, because the quality of commercial seed basically depends on the initial germination of the seed. And this is maintained either by minimum germination standards in some countries, and in others, truth in labelling. So we can guarantee that the seed we buy can germinate. But is that always the case when you get into the field? And this is going to illustrate in a different way some of the points that Gerhard made. And he was looking at a number of species and different numbers of lots, working from chickpea, alfalfa and maize to some vegetable species here and some flower species at the bottom here. And if we look at the range of normal germination, they're all commercial seed lots and all have a very high laboratory germination. But look what we see in the field. We've got field emergence of most of these crops, emergence in the glass house in modules for the flowers. But if we look at the emergence, there's a very wide range in each case. We take maize as a particular example. We've got normal germinations of above 90%, but look at the range of germination, emergence in the different seed lots. We've got a lot with an emergence as low as 48, one was as high as 98. If we look at cabbage and cauliflower, very high germinations above the minimum, but the very wide range of field emergence. And the same occurs if we look at the flower species here, high germination, but even in the glass house, a very wide range of emergence. And these differences reveal the vigour problem. We have some seeds with a high germination that emerge very poorly in the field, others with a similar high germination with a much better emergence in the field. And here we can see some picture examples of this. Here we've got Phaseolus vulgaris, and these rows here are guard rows, but we've got a block of one seed lot here, clearly emerged well, a block here that has emerged poorly. Maize, a row here and here, where we have good emergence, very poor emergence here, indicating low vigour. An example from a flower species, Tagetes, very good emergence here, very poor emergence, that's low vigour here. And it's worth pointing out that here we've got good emergence as well, but the seedlings are smaller than here. Smaller seedlings indicate a slightly lower vigour, and you'll see why later on. We can see the same problem of vigour in the glass house. Here we've got 2 trays of cabbage seeds. This one has high vigour. You can see majority of seedlings have emerged and they're quite big seedlings. Here, so at the same time, we've got low vigour and we've got gaps where some of the modules are empty and the seedlings are much smaller. In lettuce, you can see here, all these modules are very clearly filled with a seedling, everything's germinated. At the same time, here's a close-up of another module where you can see very clear empty cells, empty modules, and the seedlings that have been produced vary a lot in size. So this gives us our second physiological aspect of seed quality, which Gerhard has already identified and defined for us. But this is the sum of the properties that determine the activity and performance of seed lots, which have a high acceptable germination in a wide range of environments. Now, the impact of that low vigour varies. In the field, if you have poor emergence, that'll give a reduced yield, unless, of course, you have a cereal that can compensate and gets compensatory growth. Where the emergence is less uniform or there's variable seedling size, this gives a reduction in marketable yield, particularly in vegetables where specific sizes are required and therefore the plants are expected to be at specific spacings. For transplanted production, poor emergence obviously gives an unpredictable number of transplants produced, but also an economic effect in that you've lost the cost of heating a greenhouse, compost, staff, etc. You also get a reduction in marketable yield if the emergence is less uniform and variable seedling size. Another impact of low vigour that I haven't illustrated is that you get differences in the storage potential, and low vigour seed have poor retention of germination compared to high vigour seed. So why do we get these differences in vigour? Well, the major cause of seed differences in vigour is seed ageing, which you can illustrate on the seed survival curve. This here is the percentage germination of a seed lot over a period of time. And over that time, the germination remains high, while the seed lot's gradually aging, begins to fall a little bit, and then falls very rapidly. This is the minimum germination for sale, could be perhaps around 80% in many species. So all the seed lots on this slow decline are all acceptable for sale. But A comes early on the survival curve, B later, and C later. So as seeds move along here, they're aging. So that all the three lots have a high germination, but they differ in physiological age. A is physiologically young, C is physiologically older. And this aging can occur on the plant before harvest, which is what I call weathering, during processing, during storage, and during transport. We can use the changes that occur during this period here within vigour testing, because during aging, we can see a number of changes. The first change is membrane deterioration in living cells. And as this accumulates, you get the development of dead tissue. Now, both these will lead to an increase in solute leakage when you put the seeds into water, as the membrane integrity has been reduced. And we can use this feature in a conductivity test to determine the extent of aging and therefore the vigour of the seed. The next stage in aging is a reduction in enzyme activity. And this includes, of course, the respiratory enzymes, one of which is the dehydrogenases. Now, we can detect the activity of dehydrogenases and therefore the amount of living tissue by staining seeds using tetrazolium chloride. And where the dehydrogenases are active, you get red staining. If there's no staining, we know we've got a reduction in living tissue. This is followed then by a reduction in respiration and reduced synthesis of macromolecules and slower, less uniform germination, which is the basis of the radical emergence test. And then we can use the whole of the aging process in other tests. So we age the seeds and look at the consequence of that. So the two tests, accelerated aging and controlled deterioration, reflect the whole of the aging process. After this, of course, we begin to see the development of chromosome aberrations, abnormal seedlings, and this gives us the germination test. Now, I want to start focusing on this radical emergence test. And first of all, another activity that could be occurring at this stage, and one which Gerhard has already alluded to. And to do this, we look at the germination progress curve of maize. And this is the percentage radical emergence over the time that after the seeds are set to germinate. These 3 seed lots are all commercial lots. We've had a germination of over 90% normal seedlings. But you can see that during germination, these 2 germinate very rapidly, and this one more slowly. And this slower rate of germination is due to seed aging. But an important feature here is this lag period, the time between the start of where the seeds are set to germinate and the first appearance of the radical. It is short in the unaged seed, much longer in the aged seed. And it's suggested that during this period before radical emergence, metabolic repair takes place in the seed. And we've had evidence for repair for many years. This was first suggested in 1970 by Trevor Villiers, who stored dormant lettuce seeds. And he noted that if you store the seeds dry, they would accumulate chromosome abnormalities. If they were stored imbibed, he found no abnormalities in the seeds. And his hypothesis was that in the imbibed seed, you could see metabolic repair of membranes and DNA. So any damage that accumulated during storage was repaired in the imbibed seeds. We now have evidence of DNA repair. First of all, it was seen in the lag period before radical emergence in rye. Work was done by Daphne Osborne in the 1980s and 90s. And more recently, we've seen evidence of DNA repair in Arabidopsis mutans, which is the work of Wanda Waterworth and her colleagues. We also have some evidence for membrane repair in radish and oilseed rape. Now, in aged seeds, we've already suggested you have higher leakage, and higher leakage is associated with slower germination. And if the dry seeds are given a brief period of hydration and then dried, you find the amount of leakage is reduced, which suggests that during the very early imbibition periods, there is some repair of the membranes and therefore reduced leakage. So this brings us to the aging repair hypothesis, which we believe is the basis of vigour and vigour tests. We start with the seed survival curve, and we have differences in the position of the seed lots on this part of the curve due to aging. And we can determine the position using different reflections of aging, in accelerated aging, controlled deterioration, electrical conductivity, or tetrazolium staining. Then we can look at imbibition. This insert here shows you water uptake due to weight increase over the first 50 hours, and you see a very rapid uptake of water coming to a plateau. This is what you're seeing here. During this plateau, metabolism begins, and that includes repair. And at the end of the plateau, the radicals emerge, but the radical only emerges after repair has taken place. And then we can apply this to the germination progress curve. And you remember, you've got a short lag period for the unaged seed, and you've got a longer lag period for the aged seed. And that's because in aged seed, there's a greater need for metabolic repair. There's more deterioration. And because there's longer for repair, it's longer before radical emergence takes place, giving us the basis of the radical emergence test. So I'd like now to go into a bit more detail of how we move from the germination progress curve to the actual RE test. We've got here the germination progress curves and the different lag periods. Now, how can we assess what's going on there? And we can describe the whole of a germination curve by calculating the mean germination time. And that is based on the number of newly germinated seeds at one time, multiplied by the time, and this is summed and then divided by the total radical emergence. This gives us the mean germination time. And that's actually exactly the same thing as the lag period. So by making a calculation from the curve, we can get an estimate of the Mean germination time, that is the lag period. But that's a lot of work to do to assess seed quality. So we need a simpler way. And what we find, if you look at a single place, single time of radical emergence, you see very clear differences in the radical emergence of different seed lots, particularly clear in this example. And in maize, we found this at 66 hours. And that single count, at 66 hours, predicts the whole mean germination time. And you can see that in this graph here. This is the radical emergence after 6 hours at 20 degrees and the mean germination time. So that single count after 66 hours, where it's high, it's a low mean germination time, rapid germination, where it's a low radical emergence after 66 hours, the seed is taking a lot longer to germinate, high mean germination time. And what's more, that radical emergence after 66 hours predicts the field emergence. So high radical emergence, high RE, higher emergence in the field, low RE, lower emergence in the field. That's in maize, seen in many other species. So we can see how there are many species where a single radical emergence count predicts final emergence. These here are the 5 species where radical emergence is validated for use on the species and their ISTA rules. You can see that the timing of the radical emergence is quite short. 66 hours is the longest for maize at 20 degrees, but 30 and 48 hours here. And in each case, there is a significant relationship between RE and final emergence. I've got some examples of other species here. Again, the timing tends to be quite short, except perhaps where you've got a longer time for germination as here. But in most cases, there's a significant relationship between the RE and emergence. And since 2020, since the year 2000, there's been more than 20 species that have shown a relationship between RE and the vigour of the seeds. And the species that are include field crops, forage crops, vegetables, flowers, and grass species. So clearly, there's a very wide application of the radical emergence test. Now, that brings us through these parts of my presentation, and I'm now going to look more towards the future and the introduction of image analysis to the RE test and the potential for molecular methods. Now, one thing that happens frequently is that the industry wants to have a shorter time for involved in seed testing, either shorter tests or make them easier with automated tests. Now, the radical emergence test, the seed vigour, as illustrated already, is a short test, mostly within about 48 hours. So is there potential for automation? Well, we've looked at that using 2 methods of image analysis, with red, green, blue analysis for oilseed rape to see if it predicts vigour, and RGB and multispectral imaging of the Brassicaceae, the prediction of field and plug emergence. So we'll start off with the red, green, blue analysis with oilseed rape. And digital imaging using RGB has been used in France at the official seed testing station in Angers, ever since 2004. They do digital imaging in Jacobson incubators here. They germinate the seeds on the top of paper. It allows high throughput. There's good thermal precision and repeatability. There's 3 cameras on the top here, which can take photographs of the germinating seed every 2 hours. And here we see a typical germination progress curve from Angers for oilseed rape. And as we've seen before, you can see differences between the seed lots. Now, the earliest time here, though, we felt you could see differences in lots was at 30 hours. And at 30 hours, that single count predicts the mean germination time. So it predicts the differences in the curves that we can see with an R² of 0.91. And that single count at 30 hours also predicts vigour. So here we've got field emergence, and this is the RE assessed using RGB analysis. A high RE as a result of that analysis. We've got high early germination and high early emergence and high final emergence. Where you have a lower RE after the RGB analysis, you have a low early emergence is slower, and we have a low final emergence. So RGB is identifying RE and relating that RE to vigour. I now move on to RGB and multispectral imaging in the Brassicaceae, and this was a project which was funded by ISTA. We used commercial seed lots of these 5 species, 3 or 4 cultivars of each species, and between 10 and 13 seed lots. The first thing we did was to produce germination curves using the RGB analysis for all the seed lots. And then we looked at the germination at each time, the radical emergence each time, and we selected one time for relating to the emergence in the field and glasshouse. Now, what we're seeing here is 4 species, cauliflower, cabbage, mustard, and radish, and the field emergence. And this is the automated radical emergence count, which is at different times for the different species, 80 hours for cauliflower, 52 for cabbage, 44, and 44 for the mustard and radish. In each case, this lower line is the early emergence, the upper line is the final emergence. And you can see if cauliflower, where you have high automated radical emergence, high early emergence, high final emergence. Whereas where you a low automated RE, you have low early emergence, slow emergence, and low final emergence. And that applies for all species. High vigour, high RE, low vigour, low RE. Get high vigour, low vigour. That's in the field. In the glasshouse, not surprisingly, as you see here, the final emergence of all the species is very high, because of course we've got very good conditions for growth. But there are differences in the early emergence, which is important for producing uniform seedlings. So here with cauliflower, a high RE from the RGB, very rapid emergence, low RE, slower emergence. That is likely to give smaller, more variable seedlings. The cabbage, high RE, rapid emergence, low RE, lower. The same with mustard, and even more so with the Chinese cabbage, which emerges very quickly. So we moved on from RGB to do multispectral imaging using the Videometer Lab, which looks at uses 20 wavelengths and it takes 3 to 5 minutes for image acquisition. We're able to identify non-germinated seeds and germinated seeds. And here we have the radical emergence after 48 hours assessed using the MSI. And for orange, it's cabbage, blue, it's cauliflower. This is the mean germination time, which we've taken from the RGB germination curves. And you see very clearly for both species that there's a correlation between RE assessed by MSI and the mean germination time. So the RE is predicting differences in the whole germination curve. And if we look at the field emergence, the automated RE relates to field emergence very clearly in both cabbage and cauliflower, with high RE from MSI giving high emergence, high vigour, low RE, low emergence, and low vigour. So we can say that there is potential for the partial automation of radical emergence test, since red, green, blue analysis assesses RE in oilseed rape and predicts vigour as field emergence. And both RGB and multispectral imaging assess RE in Brassicaceae species and predict vigour as field and plug emergence. But I now think about whether there are molecular methods which could assess seed vigour. And there's two areas of research emphasis that may suggest this could be possible. The first is work on proteomics using Arabidopsis thaliana. And for this, I refer to a review by Raju et al. In 2012. And they concluded that mRNAs, stored proteins, and de novo transcription contribute to successful establishment. And this led them to suggest that proteomics and other omics studies may identify markers of seed quality. But they used aged seeds of Arabidopsis. There was no evidence from crop species. There were two instances, though, where crop species were investigated. And this was on sugar beet and alfalfa, but single seed lots were used of artificially aged seed. The other research area that perhaps suggests potential is on repair during germination of Arabidopsis by Wanderwater and her colleagues. And they first identified two ligases in the repair pathways which would remove DNA damage during germination, and concluded that ligases are the major determinants of Arabidopsis seed quality. They then went on to identify two checkpoint kinases, which activate checkpoints that would delay or stop cell cycle progression. And this would mean that growth would be delayed, facilitating repair. So there could be potential markers in this work. So to think about molecular approaches to seed testing, there has been the identification of possible markers of vigour in model species such as Arabidopsis or Medicago. But to have practical value, we've got to ask the question, do the same markers occur in commercial seed lots that differ in vigour, crop or horticultural species? And it would seem to me this would be a good idea or a good situation where you could combine the expertise of ISTA and ISSS members to investigate this possibility. But even if that work revealed differences in commercial seed lots, it's only appropriate for a vigour test if the results of that potential test relate to an expression of vigour, such as field or glasshouse emergence or storage potential. So to conclude, we believe that seed aging and metabolic repair have a major role in determining seed vigour and are the basis of seed vigour testing. We've shown that there is potential for use of both red, green, blue and multispectral imaging for radical emergence assessment, and there may be potential for molecular tests, but this possibly needs further work combining the expertise of both organisations. But to finally conclude, I'd like to give many thanks to the people involved in this work. First of all, in the image analysis project carried out at Gervais, where our member from the Vigour Committee, Takashi Shinohara, had a sabbatical time from the Tokyo University of Agriculture, and he worked at Gervais along with Marie-Hélène Wagner from France Vigour Committee and Sylvie Ducournau from the Germination Committee. Stan from the Vigour Committee was also involved, and Alina Fetchu, a student from the University of Angers, continued a lot of the work when Takashi went home. Beda and Michael helped us with the MSI work. And it's also appropriate to thank all the Vigour Committee, as I referred to many vigour tests where we've had to have the development of the test, validation and their introduction, and all the Vigour Committee have been involved in it. So with thanks to them and thanks to you for being here and listening to this presentation today. Thank you.
Thank you very much, Alison and Gerd, for your nice presentations. There are a few minutes left for some questions. maybe I start with one which I think is very interesting. And Alison knows that it will be a little bit yeah. Are there any species that radical emergence does not have a relationship with vigor?
Yeah, I can't say whether there are or not. The only thing I would say is that we've never worked with dormant species that have an element of dormancy. So we can't say in species with any dormancy that there will or will not be a relationship with vigor. But since aging is characteristic of all species, I would say I would find it hard to believe that there wouldn't be a relationship between radical emergence and vigor, but I can't say absolutely. There's also the possibility some species, the quality may always be so high, there aren't vigor differences. That's the other thing to remember. We only work with species where we know there are vigor differences. There may be species where there aren't any. Or there aren't vigor
Which are
problems.
good species on the other hand in that case.
Yeah.
Talking about wild species, Gerd, I got a question here on trees. Can the radical emergence test be used on tree species, for instance, Sitka spruce, Douglas fir, which require a pre-chill before germination?
Well, I think tests can be used, but I'm not the expert to say whether in the specific species this can be used. I think all tests must be adapted to a specific species, and Alison may comment on this maybe.
Yeah. I think where you have to have these pre-treatments, you have to work very hard on looking at the pre-treatments and the timing, and this is why we haven't worked with dormant seeds before, which require treatments like that. It's very difficult to put the two things together. We tried a little bit once with some cereals, but it's not easy. Is you say, when do you do an assessment of radical emergence? Is it during the pre-treatment time, or is it after the pre-treatment time? It takes rather even more work than a test development takes already.
Okay, I have a look for maybe one of our first questions here as well. That would go to Gerhard probably. How do the maternal environment affect seed vigor during seed development? For example, is there any evidence that temperatures during seed development influence seed vigor? What do temperatures affect to induce such an effect?
Yes, so there is some work, including in Brassica. So in Brassica, I have this in one of my slides at the bottom, the work by Chen et al. From the Penfield lab in 2021. They showed that temperature during seed maturation controls seed vigor, and they proposed that this acts through the ABA breakdown in the endosperm. So ABA is produced in the endosperm usually, and this slows down germination. And the extent to what this is degraded can be affected by temperature, and therefore a warm or cool temperature can affect germination and vigor.
Okay, I got another one here, which is interesting, and maybe go to Alison in that case. How many, not that I wouldn't give you interesting questions, Gerd. That's not the truth. But it's about
I'm not worried.
How many fields will be representative enough for correlating with the testing of the vigor in the lab?
So how many field trials would be required?
Yes.
One or two. People get worried about doing field emergence trials because they think they have to be enormous. They don't. What you want for a field emergence trial is probably using 4 replicates of 100 seeds of each seed lot, doing your best to sow your seeds in an area where you think the conditions are going to be adverse. So if there are bigger differences, you will see them. This could be done by sowing at the right time, or maybe using more than one site for your field trial. So you think, well, bad conditions might occur or poor conditions might occur in one of the two sites. So the important thing is one or two trials. If you see differences, that's good. See if it relates to your the results of your bigger test. And I would say here that encourage people that if you're trying to develop one of the validated tests for your own species, and you find that you've got conditions that relate to the emergence in the field, get in touch with the vigour committee, because it might be possible to work with the vigour committee towards validation of whatever you've developed and maybe get into the ISTA rules.
Yeah. I would like to go maybe to one of the other questions we had here. To Gerhard, thank you for the nice presentation. I missed during the initial slides that ethylene content during seed germination directly correlates with root development or inversely proportional. People need to understand that probably.
I hope so. Yeah, I mean, the slides were about P and ethylene development, and there it is that the ethylene is produced by this embryonic axis, which is a small part in a pea seed. The huge part, the cotyledons don't contribute. The peak of ethylene production is when the most seeds have radical emergence, and so there is an effect on hypocotyledon and radical growth. But in this work, not analyzed this in detail. For ethylene, I think compared to the sugar beet, the interesting point from a scientific point of view is that the dose or the amount you need, the concentration of ethylene and the sensitivity of plants is very different. At very low concentrations, you may get the opposite effect, for example, growth, and at higher concentration, you get slower growth. And in the soil, this is a reaction to the strong seed bed and the ability to grow strong through the seed bed. If the hypocotyledon root are shorter and wider and have more strength to grow. So it's not a yes or no. It depends on the concentration, and it's certainly not something on which you can build an easy vigour test because it's so variable.
Yeah, and as a chemist, I might want to add, don't put your banana next to the test if you're doing a test with ethylene.
Okay, I mean, take fruits, also not tomatoes or apples.
Bananas are probably the highest ethylene evolving species. Alison, based on current understanding, which physiological or metabolic process do you think are still underexplored in explaining why some seeds show consistently higher vigor than others, even when germination percentage is similar?
Oh gosh, particular process. Oh, I think a lot of that relates to some of the work that Gerhard's been doing. That we're talking about aging as being a major cause of differences in vigor, and some of the work that Gerhard's been talking about probably come into that. So, yeah, go ahead.
Yeah, I think so. Especially I mentioned that this phase 2, so radical is emerged, and then the pre-emergent growth in the soil. People have not done a lot of work on this from a scientific point of view. So our understanding there is far less advanced as with germination and dormancy per se. So I encourage people, younger people, of course, not me anymore, to also look at this early phase which is hidden in the soil and how it grows there. That's important.
Yeah, I think there's a good argument for sort of taking some of the fundamental work that's been done in general and taking it to look at seed material that differs in quality, so that you move from the generality to the specific.
Yes.
I've got a question to both of you. And it is, do you think that next generation of vigor improvement for smart climate seeds may relate to seed stress memory?
Lord.
Oh, I mean,
that
seed stress
goes
memory is
that goes into new seed developments, smart climate seeds.
yeah. I mean, there's loads going on in this area of memory, which is mainly then epigenetic changes triggered by experience different environments, and this may be to lead through to some other stages and may have an effect. So I think that this would be an interesting area of research in how epigenetic changes affect vigor, also dormancy and germination, but also these early growth stages. Whether this at the end leads to vigor test, I don't know. That's too early to say. First, we need to understand what is going on this level. So I'm talking about seed and seedling epigenetics in these early stages. I think this is a timely topic.
I couldn't add any more to that.
Maybe you can add to that one, Alison. Is there any method to enhance vigor as compared to lab especially in low temperature?
To enhance vigor? Well,
is there any method to enhance vigor as compared to lab, especially in low temperature?
So I'm assuming they're saying, do you want to enhance vigor so that there'll be better performance in low temperature? Yes. Well, I think any of the commercial methods that are used, like priming, would be the things that would be used to enhance the performance of low vigor seed. Or they'll enhance the performance sometimes of higher vigor seed as well. So I say any of them, of the priming methods would be ones that would enhance performance at low temperature.
Yes, I think important point is, I mean, if you work with commercial seedlings, they're already at a very high level. But if you look at land races, for example, we did some work there on germination of pepper and sorted these in different fractions. And interestingly, there was this fraction which could not be improved by priming for temperature responses. There was another fraction which could be improved by priming for temperature responses. And then there was a third fraction which was, I would say, hopeless. So priming doesn't think. So you better discard. So you saw
it all depends on the initial quality of the seed. They say your different fractions, the seed quality will vary.
Exactly.
Now getting a little bit special.
Oh dear.
In those species as orchids, where protocorm is the first part emerged, radical emergence should be replaced by protocorm emergency?
I think orchids are very interesting and very special. So I cannot give practical advice there, but I think this is one of the cases where you have to leave primary school and pass it first. Primary school means you look at the radical emergence. There are species where radical emergence is not the best visible sign of germination, and you simply count visible events like protoderm differentiation or formation of rhizoids and so on. So I think this is what you can do there. Whether this is Useful in practice, I don't know. I don't know what you know something about that.
No.
Practical
I
aspects
think
of orchards.
orchid seeds are such a specialised case that you're not going to be using the radical emergence clearly, but we're talking in general terms. I don't think orchids would come under the category general. They are a very special case in many different ways.
Good. We have an awful lot of questions, and we will not be able to answer them in the next 4 minutes, but you will get answers, and we will forward the questions to the lecturers, and you will probably get answers then pretty soon, I say, not during the next minutes.
The next days.
The next days, yes. And we don't even promise directly before Christmas. We will see how that would work.
Well, maybe that.
Maybe that, but
maybe not.
You didn't see the number of questions yet, Alison.
Okay. The top of the list before Christmas, the bottom of the list after Christmas.
What factors affect seed vigor during seed development, or factors to consider to get high vigor seed from plants?
Who takes that?
That's more a breeding question, probably.
Well, I could say in general that the temperature and humidity of the seed towards the end of seed production are very important in affecting seed quality. If the temperature and humidity are high, and particularly if the moisture is high, then dry, then high again, that can affect the seed quality. That's towards the end of the seed production phase. I'm not sure about very early on. I don't know if that's something that the seed development that Gerhard would know about.
No, I think you're absolutely right. I think it's the late maturation phase where the effects come in, and the same is with dormancy. I mean, depending on the temperature, you can get also dormancy or no dormancy. And essentially, the best seed quality is when all the dormancy is absent and all the vigor is highest. This is usually considered to be at this physiological maturity of seeds.
Coming back a little bit to ethylene, does seed treatment with ethylene in gourds help increase root growth and soil germination?
I mean, it does not necessarily increase root growth in length, at least not in higher concentration, but it increases or promotes germination in many cases. And in root growth, it's needed for getting through stronger soil. So it's strong plants. It doesn't mean necessarily faster. It just means stronger and able to get through. In contrast to ABA and auxin, where if this would be low, then the root growth is better in strong soil. But ethylene signaling is needed for a strong pre-emergence root and hypocotyl growth in soil.
Good. Alison, are there differences between treated and untreated seeds for the same temperature, and can we apply radical emergence test on treated seeds?
We would usually advise not using treated seed, but if you're only going to be using treated seed in your own lab, you could still make comparisons. But if you're going to use, if you're going to use a treated seed of some lots and non-treated of the others, that wouldn't be a sensible idea. That's basically.
I had another question what I would like to answer with your help, Alison, probably. Are there any vigor workshops upcoming? At the moment, I can say I don't see them directly on the line, but you maybe have more information.
Yeah. Well, we only finished one last weekend. There is a discussion about a bigger workshop. I'm not sure what year, whether it's 26 or 27 in India.
Yes,
so that is
maybe others upcoming, and I would like to invite because that came from Korea and Italy, like to invite also laboratories, especially in Europe, to register and approach the Secretariat or the vigour committee on vigour workshops to be hosted by themselves. That is something we always look into, and we always highly appreciate because we need to hold these workshops, and good preparation of a lab is very important on that.
Well, there must be a good interest in Italy, because we had 4 people from Italy at our workshop last week.
Yes, it seems to be. I thank both of you. Those questions we did not answer, we try to answer later. Don't forget, you can get all the presentation, our presentation on the ISTA YouTube channel. There was a link in the chat for that, and a lot of other links were in the chat as well. So thank you very much, Alison, Gerhard, for your presentation. And I wish everybody happy holidays or Merry Christmas wherever you are in the world, and whatever you prefer. Have a good time at the end of the year. And thank you to both of you again, and to ISSS and ISTA in that case, to arrange these series of webinars. Thank you very much. Take care. Goodbye.
Thank you.
Goodbye.
Goodbye.